Let's discuss about Cartagena protocol on biosafety. Before that, let's recap a bit from our previous videos. In 1992, the Earth Summit, the Rio Earth Summit was organized, and in that summit, three important conventions were opened for signing. These include UNFCCC, that is the United Nations Framework Convention on Climate Change, and the Convention on Biological Diversity, and Convention to Combat Desertification. So these three conventions are called Rio Conventions, and under Rio Summit, these three conventions were opened for signing. So under Convention on Biological Diversity, we got two important protocols. These are Cartagena Protocol on Biosafety and Nagoya Protocol on access to genetic resources and benefit sharing. So in this video, let's discuss about Cartagena Protocol on Biosafety. And I want to clarify one thing here, that a convention is a forum through which various countries meet together and decide on a particular issue. Here it is the Convention on Biological Diversity. Here in this convention, the biological diversity is discussed. So only broad framework on how best the biodiversity must be conserved is decided in this convention, and the member countries did not go deep into the aspect of the procedures, rules and regulations for conservation of biological diversity. So this is left to the task of the member countries, that is the conference of parties. The representative body of the member countries are called conference of parties. They meet regularly to appraise the situation as well as to undertake decisions to bring the objectives of the Convention on Biological Diversity. So this conference of parties meets regularly, and in 2000 this conference of parties decided to establish Cartagena Protocol on Biosafety. So this protocol lays down detailed rules and regulations, procedures that must be followed while transboundary movement of certain genetically modified organisms to protect the biodiversity. Okay, let's discuss about this. Here, the Cartagena Protocol on Biosafety is the first international regulatory framework. So it is the first international regulatory framework for the safe transfer, handling and use of living modified organisms. So this protocol will regulate the safe transfer, handling and transboundary movement of genetically modified organisms in order to protect the biodiversity. So this protocol was agreed under Convention on Biological Diversity, as I discussed earlier. And this biosafety protocol seeks to protect biological diversity from the potential risks posed by the genetically modified organism result from modern biotechnology. So these genetically modified organisms were produced with the utilization of techniques from modern biotechnology. So this biosafety protocol makes clear that the products from the new technologies must be based on precautionary principle. What is this precautionary principle? It allows the developing nations to balance public health against the economic benefit. So on the one side public health, on the other side economic benefit must be balanced. Okay, there must be a balance. There should not be precedence for one thing or the other. Okay, these two must be balanced. And this protocol has 170 parties, that is the member countries, which include 167 United Nations member countries and also 3 non-UN member countries. These include NAU, State of Palestine and European Union. And United States is outside this protocol because it has not ratified the Convention on Biological Diversity. Here, the protocol defined various terms which are relevant to the convention. These include living modified organisms. These living modified organisms are defined as the organisms that possess a novel combination of genetic material obtained through use of modern biotechnology. See, what is this novel combination of genetic material here? Here, novel combination means, take for the example of Bt cotton. So Bt cotton is a genetic modified organism. This Bt cotton was produced by the procedure of inserting a gene called Bacillus thuringiensis gene. It is obtained from soil bacterium, and this gene is inserted into the Bt cotton, okay, in order to provide resistance for the crop towards certain insects. Okay, after inserting this Bt Bacillus thuringiensis gene into the cotton, this cotton will get tolerance, that is resistance towards the insect called bollworm. So this bollworm feeds on the cotton ball, okay, the fruit of the cotton, okay, and it is very devastating insect. So after inserting this, the whole plant will become poisonous for this bollworm, so that after feeding on the cotton ball, it gets killed. So this is the mechanism here. So this is this plant this product is obtained through novel combination of genetic material, because the Bt gene is inserted into the normal plant. So this is novel combination using modern biotechnology. So genetic recombination is used in the production of this plant. So this is the meaning of this. And the living organisms means any biological entity capable of transferring or replicating genetic material. Anything, any organism, including the sterile organisms like virus and virids. And here the modern biotechnology is defined in the protocol, means the application of in vitro nucleic acid techniques. Certain techniques are used in the biotechnology. These include in vitro nucleic acid techniques, fusion of cells beyond the taxonomic family that overcome natural physiological, reproductive, or recombination barriers, and are not techniques used in traditional breeding and selection. Here, the techniques which are not used in traditional breeding and selection methods, and also including novel techniques like nucleic acid techniques, in vitro nucleic acid techniques, fusion of cells beyond taxonomic family. Here, what is the meaning of this fusion of cells beyond taxonomic family? Because naturally there is a hierarchy of plants. Okay, these are classified under taxonomic family. And here take the example of Bt cotton itself. This Bacillus thuringiensis gene is not present in any of the natural taxonomic family of the cotton. Okay, it is a novel feature. It is an artificial feature added through modern biotechnology. So this is called beyond the taxonomic family through use of modern biotechnology. Okay, and let's look at with the diagrammatic representation here. What is genetic modified organism? Here, take for example a plant. Take for instance, it is a brinjal. Okay, if you insert a desired gene from other organism, maybe a plant or animal or a microorganism. Okay, if you insert from that source a gene through a carrier, this is the vector called. It's called vector. Usually in biotechnology we use agrobacterium. It's a bacteria which carries genes into the desired plant. Okay, and if you insert the desired gene into this bacterium and transfer this gene to the desired plant by means of DNA recombination technology, okay, then it will result in genetic modified organism. Okay, here the crop, if you insert unrelated gene to that crop, then it leads to genetically modified organism. So there are a lot of genetic modified organisms present in the world today. These include various animals, microorganisms, plants. So various living organisms are modified through use of modern biotechnology. And I am going to give you some plant sources of genetically modified organisms. These include alfalfa. So as an important gene was added to this plant in order to provide the resistance to the glyphosate. Okay, here glyphosate is a herbicide, weedicide, which is a chemical used to kill the weeds. Okay, and by inserting a certain gene, this plant alfalfa crop is made tolerant to this glyphosate. Thereby it can be applied, this glyphosate, that is the weedicide, can be applied in the field to control weeds which grow in between these alfalfa plants. Thereby it will only kill the weeds and it will not kill the alfalfa plant, because it gets resistance because you inserted a gene which makes the alfalfa tolerant to this glyphosate plant. So this helps in reducing the cost of cultivation of the alfalfa plant, because you can indiscriminately apply the chemical without concerning the safety of the alfalfa plant, because it is resistant to glyphosate. So previously what happened, the farmers used to take up manual weeding. That means by hand they used to pull up the weeds. It involves huge cost. But here only they indiscriminately spray the glyphosate in order to kill the weeds. So this is an economy in the agriculture. So this is an advantage. And here in apple, there's a physiological disorder called delayed browning. So the browning of apple happens. So if a certain gene is added, it reduces the polyphenol oxidase in the apple, thereby reducing the browning. Okay. And rapeseed, in rapeseed also tolerance to glyphosate was made with the addition of certain gene through modern biotechnology. In cotton, the most best example of a genetic modified organism is the BT cotton. So Bacillus thuringiensis, as I discussed earlier, is a soil bacterium. Okay, soil bacterium. It is a bacteria, and a gene is present in this bacteria which help to control certain insects. Okay, it has insecticidal property which kills the insects. So this cry protein, cry protein gene is added to the cotton through biotechnology, means thereby it provides resistance to certain insects, because after adding this gene the cotton plants gets resistance, thereby it becomes poisonous to the insect itself. If an insect feeds on the cotton plant, that is a BT cotton plant, it gets killed because of the presence of BT gene, Bacillus thuringiensis gene. So this GMO, genetic modified organisms, result in 49% change in the area. Okay, 49% of the traditional area is changed to the genetic modified organism. Okay, and another example is BT brinjal. So brinjal has certain insects that feeds on them. This include the fruit borer. Okay, so insects bores on the fruit, thereby damaging, and as well as shoot borer. So insect also burrows inside the shoots. So in order to control this, a gene, that is BT gene, is added to the brinjal plant, thereby making the whole plant poisonous for that insect, thereby protecting the plant from the insect damage. So this is also one example of genetic modified organism. In papaya, papaya has a serious disease called papaya ring spot virus. So in order to control, in order to provide resistance to this disease to the papaya plant, a gene is added. This is also genetic modified organism. And in the rice, in the rice, the certain gene is added from maize. Okay, from maize, gene is added to the rice, and it helps in enriching the beta carotene, vitamin A of the rice. Okay, this is called golden rice. So inserting gene from the maize and a common soil microorganism to the rice enables rice to enhance its nutritional capability, thereby it may in the future help in reducing the malnutrition. So these are all the works that are done in the genetic modified organism. And why there is a threat to the biodiversity by using living modified organism and genetic modified organism, which is the main aim of the Cartagena protocol on biosafety, to regulate the movement of these organisms. So here there are certain threats to biodiversity here. If you use genetic modified crops, it may lead to horizontal gene transfer. For example, if BT cotton or BT brinjal plant waste is fed by the animal, it may lead to horizontal gene transfer from that animal source to the plant source. So genes may be transferred artificially. Okay, and there are a lot of animals that feed on the plant sources. Okay, for example, cattle. If cattle feeds on the BT brinjal or BT cotton, what happens to their health? So since this plants carries poisonous genes, that is the Bacillus thuringiensis gene. So this is another problem here, because most of the developing countries has animal rearing as the main occupation of the farmers. So the BT maize and BT brinjal has Bacillus thuringiensis gene in them, and these are poisonous to the insects. And what happens if it is poisonous to the human beings as well, by means of biomagnification or other means? Okay, this is another concern here. And here, with related to environment, as I discussed earlier, BT, that is the Bacillus thuringiensis, is a soil bacterium. It has two important proteins. These are called genes which produce protein. These are called cry protein and VIP protein, VIP protein. These proteins are toxic to the pests. So this, if genes from this plant is from this bacterium is inserted to a plant, then it brings benefit to the plants, thereby protecting them from pest attack. And there are other concerns that by utilizing more and more genetic modified organism, it may crowding out the local varieties. So it may reduce the use of local varieties which have other advantages as well. Some of the local varieties are more nutritionally rich, as well as they have more drought tolerance. Okay, they have other important capabilities. If you use more genetic modified organism, it may lead to crowding out of these local varieties. This is one of the biodiversity threat. And another concern is that of cross pollination, because these genetic modified organisms are a class of plants or animals which are distinctly different from the natural plants. So if this in plants we have cross pollination. Okay, pollination will occur from one plant to the another. So if a cross pollination happen between the genetically modified crops and a normal crop, it may lead to genetic impurity of the normal crop, because it adds the genetic modified gene into the crop. So it may bring genetic impurity in the ecosystem, thereby destabilizing the natural balance. And another concern is that uh, we have these geographical indicators. We have basmati rice. Basmati rice are worldwide famous because of their flavor and their distinctive uh, area of growth in India. Okay, India and Pakistan. If we develop genetic modified rice, if there is a cross pollination between basmati rice and genetic modified rice, then it lead to a worldwide reduction in demand for this basmati rice, because its genetic purity is violated because of its crossing with the genetic modified rice. So this is another apprehension here. And this this genetic modified crops also may lead to super weeds. What is this super weed? We all know that in our previous slide we discussed about the tolerance to herbicides, glyphosates. So these plants may become unable to control in the wild. It may grow as wherever it likes, because of the resistance to certain weedicides. So it may lead to development of super weeds. And even if you use bt gene into the cotton or other important plants like maize and brinjal, even if you control the major pests like bollworm and other things, secondary pests, other pests may also emerge, and secondary weeds may also emerge in the event. So nature always try to overcome the human barriers. If you control one pest, other pests may emerge in the natural environment. So this is not the solution. So this is the thing here. So this is about genetic modified crop I have already discussed. So the article, the protocol also discusses about precautionary approach. So Rio Declaration on Environment and Development in Earth Summit, Rio Declaration was made. It contains 27 principles, out of which the 15th principle states that in order to protect the environment, the precautionary approach shall be widely applied by the states according to their capabilities. So state must ensure the precautionary approach. Okay, where there are threats of serious or irreversible damage, lack of full scientific certainty shall not be used as a reason for postponing cost-effective measures to prevent environment degradation. So this is the precautionary principle. And where this Cartagena protocol is applicable, here the protocol applies to the transboundary movement. So if there is a movement of living modified organism or genetic modified organism from one country to the another, and also transit from one boundary to another, handling and use of all living modified organisms that may adversely affect on the conservation and sustainable use of biological diversity, taking also into account risks to human health. So this Cartagena protocol biosafety is applicable to transboundary movement, transit, handling, safe handling, and use of living modified organisms. So various rules and regulations are laid down under this protocol to regulate these living modified organisms in order to protect them from to protect them not to harm the biological diversity. So important features included in this protocol is that overview feature. This overview feature, the protocol promotes biosafety by establishing rules and procedures for safe transfer, handling, and use of LMOs, as I discussed earlier. And other important agreement under the protocol is Advanced Information Agreement. Okay, this Advanced Information Agreement is a procedure applies to the first international transboundary movement. Whenever there is a first international transboundary movement of any living modified organism or genetic modified organism happens, this Advanced Information Agreement is applied. Okay, for excuse me, LMOs for international introduction into environment of the party of import. So if India wants to import a genetically modified organism for the first time, okay, from US, then this advance information agreement will come into force. Here the United States has to notify Indian authority about the export of this genetic modified organism. In the meantime, India has to provide consent, approve the export of this organism, so that it will bring certainty in the information sharing of the genetic modified organism, so that it protects the importing countries by taking precautionary approach while dealing this genetic modified organism. And another agreement is that LMOs intended for food, feed, and for processing. So genetic modified organisms intended for food as well as animal feed and food processing are also included in this LMO FFP agreement. Here it is not as stringent as advanced information agreement. Here LMO FFP for the transfer of food, feed, or food processing LMOs, the countries must in advance inform the biosafety clearing house about the transaction, that is in 14 days before they have to intimate this biosafety clearing house. So this is another agreement here. And the protocol also deals with the handling, transport, packaging, and identification of the living modified organism, because handling of the living modified organisms is very much essential, because if it is not properly handled, it may lead to the hazards, the biodiversity hazards, and while transporting, certain precaution must be taken. For example, the genetically modified microorganisms, if it spills into the atmosphere, it may cause devastation, and proper packaging is specified under this protocol as well, and as well as identification through proper labeling. Okay, the genetic modified organism in the transboundary movement must be labeled properly, including the source, okay, origin, date of packaging, and source of country and destination country, like that. Okay, and the protocol also mentioned about biosafety clearinghouse. So it is an institution. Okay, the protocol establishes biosafety clearinghouse in order to facilitate the exchange of scientific, scientific, technical, environmental, and legal information. So it facilitates the exchange of these information, including scientific, technical, environmental, and legal information on living modified organisms. So it helps in assisting the parties to implement the protocol. See this biosafety clearinghouse. So this is it about Cartagena protocol on biosafety. So thank you for watching. Please share the video.